So good afternoon, everybody. I'm Stefan Stoll, and on behalf of the department, I would like to welcome all of you to this year's Paul C. Cross Lecture. We have a fantastic speaker here, Songi Han from Northwestern University. Well, this lecture is named after Paul C. Cross, who is, let's see if it can disappear. Who was the chair of this department from 1949 to 1961. Paul C. Cross got trained in Geneva College and University of Wisconsin, and then was postdoc at Wisconsin, Caltech, and Stanford. He became an assistant professor at Stanford for a few years, then moved as an associate professor to Brown. And then finally, he came to the University of Washington to head and to lead the Department of Chemistry. Under his leadership, the chemistry department grew from 18 faculty to 24, which doesn't sound that impressive, but one has to consider that during his tenure, the Department of Chemistry split off the biochemistry department, and it also split off the chemical engineering department. So the net gain was substantial. So he has grown our department quite a lot. And along with the faculty count, also the graduate program. He's one of the people responsible that the University of Washington really established itself as a top tier research program in the country. He left University of Washington to become president of the Mellon Institute. And under his time there, that Mellon Institute merged with the Carnegie Institute of Technology to become Carnegie Mellon University. So this Paul C. Cross lectureship is dedicated to his memory by his family and his many friends and colleagues. And we're very grateful for the continued support to have this cross lectureship going. Now we have a long list of very established and excellent speakers here, starting in 1980. There's a few gap years, including 2020, as you can see. And so today, for 2024, I'd like to introduce to you Professor Songi Han. She got her training, including her PhD in Germany at the Aachen University of Technology, and then she moved to UC Berkeley as a postdoc. And then she started her independent lab at UC Santa Barbara, while she over the years moved through the ranks to become full professor. And as of last year, she is now the Mark and Nancy Ratner Professor of Chemistry at Northwestern University. She has a very vigorous, innovative research program that touches a lot of different fields. And she has received a ton of awards. And a few selections are shown here on the slide. NSF Early Career Awards, Packard Fellowship Prize for Teacher Scholar, an NIH Innovator Award, a Bessel Humboldt Prize from the Alexander Humboldt Society, Innovation Award from the Biophysical Society, and one of the more recent ones, the Bruker Prize from the Royal Society of Chemistry. So we're super excited to have her here today to tell us some of her extensive research. And I just briefly summarized that for you. She's worked on using magnetic resonance spectroscopy predominantly to study surface water and structure, and we will hear about that today. She's looking at biomolecular interactions. She's worked on tau protein aggregation, so at the molecular basis of neurodegenerative diseases. She studied the fundamentals of dynamic nuclear polarization. And if you're interested in that, I encourage you to come to the physical chemistry seminar tomorrow. Well, she will present more details about that. And she, in order to help with all these innovative endeavors, she's also developing new technology for better measurements. So without further ado, I'd like to ask Professor Han come forward so I can give her a little piece of lumber from Washington State. Congratulations, and thank you for coming.
Thank you very much.
You turn on the microphone. All right, thank you very much.
Thank you, Stefan, and it's a great honor to be giving the Cross Prize Lecture. I had today already a really exciting day with each conversation. We ran over because there's such a good community, and you know, I could tell from the environment that this is a great place to be, to do research and for the students. So thank you very much for having me. So I'll be talking about the shape of water, and hopefully through the lecture, it will become clear what do I mean by that. So let me see how do I, and then, oh, maybe that's how it, okay. I'll just, okay, it'll work. So the shape of water on active surfaces, and you all know water, and I think many of you love water. And I just want to remind you, water is the most unusual molecule. It can do many things, but among others, it has the ability to form various structures. I mean, we all know that it can have two hydrogen bond formation. If the angle of the HOH is 109.4 tetrahedral, that's when we have ice. But we also have other angles, such as 60-degree angles included in liquid water. And we have a myriad of possibilities for water to form organized networks. Water is also a field that is very exciting and very dangerous in a sense that there's so much controversy out there because it's an exciting substance, but also it's not easy to measure. So that's where we hope we have made contribution in developing magnetic resonance-based techniques that help us interrogate this fascinating material. So we all agree when I say from the left to right, it gets more complicated. Of course, water stabilizes a 3D protein structure. You would all say that's obvious and true. We also know, and the second portion would also be not as controversial if I said the change in water, either structure, property, or content, mediates conformational changes. You also would agree that there are many examples. But see how the word becomes increasingly active, mediates. Next one, and that's what I would like to tell you about, local water structuring also shapes what we will refer to as a hotspot to guide assembly. So that's going to be the topic I'll focus today. And the part that I will probably not get to is how even more actively the change in cohesive or coherent domains of water movement can drive protein action. So it goes from more passive all the way to active. But we teach our students whenever they practice their talks, don't give two stories, don't squeeze two talks into one. So I will focus on the story of assembly, but let me first make one thing very clear before I go into my story. So in the more passive version, it's not so obvious that we need multiple structures of water because in the case when I say changes of hydration can mediate conformational changes, well, it could just be water is hydrated at the crevices and squeezing out of it could induce conformational changes. But as soon as I talk about local structuring of a so-called hotspot, and it will become clear what I mean by that is, it becomes clear that we have to have at least two populations of water at the minimum for this differentiation to occur. Or even more when I say certain proteins could act like a hydraulic pump, then it's not enough to have one kind of water. We need at least two kinds of water, the one that is more agile versus the one that stabilizes the protein. So the story will be really evolving this idea. And here I'm just showing you a calculated plot of bulk water measuring the three-body angle distribution of the HOH angle and how it's distributed in bulk water. And you see that the angle distribution aren't just randomly distributed relative to bulk, but we have distinct peaks at around 60 degree and around 109. The 109 is the tetrahedral water, and the 60 degree is close packing icosahedral water. And a different coloration is bulk in this particular case as we increase glycerol concentration. But this sort of a pattern is seen with many or most solutes. Okay, So I'm not saying there's just two structures of water, but at the minimum, we have different packing, such as tetrahedral and icosahedral packing. Okay? So the idea of ice-like water, a decade ago, when I went to golden continents of water, was almost a forbidden term. People said, oh, this ice-like water, that's a misunderstanding. It turns out by now, it's not so wrong to say ice-like water, not in a sense of not in a sense of dynamics, but in a sense of, for example, a three-body angle. There is such a thing as ice-like water. And my story hopefully will make it clear why this is so important. So I would like to mainly focus on the story, how we have to understand as well as utilize structuring of water to achieve templated self-assembly. I decided to focus on this story because I can tell you the science story while better telling you about the tools that we needed to develop to really get at the core question. How do we fold and control the shape and assembly of this important protein tau? I couldn't help it but to still flash the second topic that I will only at the end conclude, namely our discovery that flavonoid photoactive protein seems to be really driven by the action of water. So I'll at least conclude, but not go through the story. Okay. So let me give you a little bit of context so that it's clear why is it important to even think about templated self-assembly and why do we even need to do that, right? So the context of neurodegenerative diseases is that many of them, or vast majority of them, are proteinopathies. That doesn't mean that the cause of it are these proteins, for example, alpha-synuclein for Parkinson's, or tau aggregation for a myriad of diseases. About the cause, there is a lot of debate, but that's at the end a hallmark. That's what we see at the end. That's how diagnosis is done. Another important point is that these tau proteins spread in a prion-like fashion. So the tau protein, which does underlie Alzheimer's disease, CT, and a myriad of neurodegenerative diseases are called prion-like proteinopathies. So a breakthrough, a very, very important breakthrough was made around 2017 and 18. People have been studying tau aggregation for a long time. But only when the cryo-EM structure came out and then this Alzheimer's disease structure from a patient came out, I remember being in a FASEP meeting and people gasped because the structure that was solved was completely different from what people had imagined the tau structure to be. Many careers were destroyed actually trying to solve the tau structure because it turns out only in the human brain are these structures super well defined. And whenever you try to generate it in a laboratory, it's very difficult to make them homogeneous. So imagine crystallography, NMR, all sorts of other techniques with synthetic fibrils went nowhere. But in the brain material, it's well organized. So the most important discovery is that for each disease, n equal 10 in the case of Alzheimer's, n equal 100 meanwhile, or hundreds. It's always the same structure. Pick's disease is always the same structure, etc. So independent of the cause, at the end, it's the diagnosis criteria. Now, why am I drawing these arrows? I'm drawing these arrows to kind of emphasize that the same tau molecule fold into different conformation and they stack. And at a molecular level, they have distinct structure as can be measured even by pairwise distances. And I'll come back to that point. Okay? But a very important point I would like to make is we know from cryo-EM now that these very intricate protein structures deposited in the brain is a hallmark. It's the diagnosis. But the current state of the clinical research is as follows. There is no early diagnosis. There is no late diagnosis. Even at the late stage, diagnosis made by interviews, not by physical measurement, at least for tauopathies. And very importantly, one of the reasons why there is not even a late diagnosis is there is no disease-specific antibody. It's a pretty strong statement I'm making, but I know that this is a true statement. There are antibodies that claim to be tauopathy specific, and some of them are perhaps AD specific, but they're not rigorously verified, and there are no imaging tool and so on and so on, okay? So the grand challenge is to now synthetically replicate. Cryo-EM gave us the structures, but those are from human diseased patient material. Those materials cannot be used as the sample basis to develop tools. So we need the knowledge base to synthetically replicate this intricate structure, and ideally in a disease-specific way, or even if not disease-specific, at least generate relevant fiber. But that cannot be done. And if there was success, they were accidental success. And the problem with accidental success is they cannot be replicated. For example, in vitro, that doesn't mean that that success can be repeated in cell or in the mouse, etc. There is no good mouse model either. So another question is, it's a grand challenge. Why should my group work on it? It turns out it's a protein folding problem. And it turns out we who thought about water a lot could contribute perhaps in an important way. So the question can be simplified into how do you fold spaghetti into a very intricate, highly defined architecture that's also self-replicating. Why self-replicating? Because I said they have prion properties. It's not good enough to have a well-defined structure. They need to be active because that's the core property, okay? So how do we solve this? So I think the next one is a cartoon. Oh, okay. So that one is the photo. So if you think about intrinsically disordered protein as represented with this random wires, it's really not easy to imagine how would I not only fold them very intricately in 2 dimensions, but a 2-dimensional structure have to forever replicate, because in order to have prion property, you need to have the fold replicate forever in register. It's a pretty stringent criteria, even more stringent than for a normal protein that folds in 3D. There you just have to fold it into one structure. Here it's a 2-dimensional structure, has to replicate. So how do we solve this issue? We need tricks, and we thought about, we can come up with 2 tricks or 2 hypotheses how to solve it, and here are the ideas. So we decided when something as intricate as this cross-section shows folds, and we are talking about 200 to 400 residue proteins that are initially intrinsically disordered, there got to be a nucleating shape. It cannot be that they fold in 2D at once, right? There has to be a nucleating shape. And then what I will show is, even if you have defined or hypothesized which shape might fold first, even that's not enough. You still need a much more localized pinning site that would hold the wires together first so that the rest can stack. So I'll talk about first how part 1, generating a nucleating core, worked. And then I'll be able to come to my main story, how we need to think about water to assemble them in an organized way. So I'll spend maybe 5 minutes on showing how this nucleating core did do what we were hoping it would do. So we took from this different disease fold, different fragments. So I have one that I circled in purple. That is the shape from a disease called CBD, corticobasal degeneration. And long story short, we were able to generate fibrils, determine our first ever, maybe last, I don't know, cryo-EM structure, My student did that and were able to show that they have prion-like properties. They self-propagate forever, they're very competent, and they can even recruit larger tau. So I say it looks like a prion, it quacks like a prion, and probably is a prion. And furthermore, not only do they do that in vitro, but they also infect cells in an isoform-specific way. So we check the boxes. I'll show you one more, and the only reason why I show you one more is because from this mini tau, we weren't able to then nucleate the larger fibers yet. We are still working on it. So let me show you just one more, because there we were able to make more progress. We then did a similar exercise on a different fragment from Alzheimer's disease. So same idea, we take that peptide, we made a chimera by connecting the loop a little bit, and same exercise, we were able to show that they form fibrils very vigorously and have prion-like property. They're very competent, and they do that in vitro as well as in the cell. In this case, we didn't do the cryo-EM, but in collaboration with Dan Southworth, my collaborator, we are able to show, and the refinement is going on, that the shape that this little peptide forms is indeed the shape that we had predicted and hoped that we would make as found in Alzheimer's fibrils. So we made a mini prion, so to speak, and it acts like that. Now, let me show just a little bit more, because I started the story by saying recreating the full fiber is a grand challenge. So let me show you our effort towards that goal, just so that you get an idea. So once we have the mini tau prion, of course, the idea is that that nucleates the full structure. So now we need to be able to take that fiber, incubate a much larger tau. We used full length as well as a slightly shorter sequence called DGAE. I think the details aren't so important. It does encompass the entire 80 fiber forming segment. And we're able to show that they fold into very good looking fibers that have prion properties. That's not completely obvious. And that they do affect infect cells. So now comes a little bit of EPR, okay? Those who have done cryo-EM will know that it's not that you can have like intermediate success and then you can work your way up. Oftentimes, the problem is it just doesn't make the right shape. It is not of enough high quality. You don't know what you have until you have the fiber that give you the quality. There is, so here in this context, it's very helpful to have structural biology tools that are able to capture the entire ensemble. So that's where double electron resonance, the EPR tools that measure pairwise distance are so powerful. And Stefan being here, I have to show a little bit of DIA, okay? So the idea is this. What I'm showing you here is a computed distance distribution of sites across 373, 351. It's a single pair. But because we know that if you have 80 fiber, CT fiber, or known intermediate structure, they're very distinct. So even a pairwise distance gives us very unique signature. Okay? That's very powerful because DIA can, in principle, capture the whole ensemble even of a spaghetti. You will see a very broad Gaussian. And if you made all three, you would see that. If you made two of them, you would see that. If it converged, you see that. So we are able to use this kind of structural biology tool to learn what do we have to do to shift the balance. So I'll show you. This is the theory, not experiment. The experiment, in my opinion, looks pretty stunning. This is the experiment. And what is stunning about it is that the three features that we thought could occur when we seed the tau with the mini-AD, namely the 80-fold, CT-fold, and the intermediate fold, all show up. But when we do the process under conditions that are known to favor AD, but it's not usually very reproducible unless we have a catalyst like we propose mini AD, then in the green one, you see that the balance shifts dramatically towards Alzheimer's disease structure. And when we then do multiple generations of seeding and just look at maybe the purple one, you see that how much we were able to shift the population towards an AD population. We think with this optimization, we are very close to being able to regenerate with a synthetic fiber cryo-EM structure. My colleague Dan Southworth is working on it. And we think we are very close to achieving what we set out to do. And all of that was still the prelude to say, we know that our little fragment idea works, but even if you take a 20 amino acid fragment and you just want to make a U shape, and then you want to stack the U shape, even that's not so easy. And I would argue that if you have an intrinsically disordered peptide and you want them to precisely stack in register, how do you do that? What sort of interaction do we need to achieve that? Okay. And whatever the trick is, it must be ingrained in at a monomer level, because this peptide can aggregate on their own, certain condition, and they make these beautiful fibers, and then they self-replicate. So what is the trick? We think there has to be a property ingrained at the monomer level that makes the peptide be attracted to a specific site. And the only mechanism I can think of that peptide that are intrinsically disordered find each other to exactly come in register. They have to really like the interaction with the same site, cannot be electrostatic. They would be repelling each other, would be some sort of hydrophobic interaction, but mediated by water. So let me explain what I mean. We have an intrinsically disordered peptide. If you have a certain site that is hydrated by structured water, and I'll qualify what I mean by that. I kind of hinted at the tetrahedrality. Then those water cluster, let's say, will be attractive to each other because they are structure match. Think of it like ice nucleation. If that was the case, this property should be measurable at the monomer level. And then once you have a nucleating fibril, then this could also, the same principle could apply when a monomer docks to fiber. But it has to start at the monomer levels, right? And at the end, the fiber, I can tell you, that's the beauty of EPI. We can measure the whole ensemble. They all make fiber. We are talking about at the end, 80% of the population make this in register fibers, right? So, here's a cartoon where I'm trying to then explain just in a slightly different way. If I have a structured water that is low entropy water, because it's forming over a small hydrophobic, less than a nanometer hydrophobic site, then as soon as it has the opportunity, because a ligand or another site comes, it can get evicted in favor of the interaction that it will form, and therefore it would be an entropy-driven association. It's a well-known mechanism, but not many experimental ways to really observe that. This is not a concept that I have invented, of course. A lot of discussions about that, about the length scales dependent of hydrophobic hydration. So I'll just contrast the two extreme cases. In one case, when I have a hydrophobe that's less than a nanometer, water cannot hydrogen bond directly to this yellow spot. What will it do? It will wrap over. It will make its own dome with its own hydrogen bonding capability. And that structure will tend to be tetrahedral because that's what it does best unless there is something to anchor. But when I have a Teflon surface, well, there's nothing to reach over, so it's longer than a nanometer. It will just basically have this very unstructured dynamic hydration. Okay? And one important thing I should mention is, what is a hydrophobic site? Water decides that. It's not necessarily hydrophobic as in oily. It can be a charged group being hydrophobic because the determinant is whether water in the context can directly hydrogen bond or not. If you're on a surface where everything is, say, positively charged, and now suddenly it has to make a turn to hydrate an oppositely charged site, that site can be hydrophobic because water can't turn its corner that fast. So for whatever reason, if water cannot hydrogen bond to that site directly, it's a hydrophobic site. So then the question is, okay, how do we measure that? How do we experimentally measure specifically the structured water? Because that's the more interesting water, right? Because it's the water that is structured, it's the water that has low entropy, it's the water that can give us the free energy back, and so on and so on. It's this agile water that we think is very important for protein activity. So there are two methods that we have developed and used over the years to get at these properties. One is local water dynamics. And I don't have time to go into the detailed technique, but I'll just describe it briefly, okay? We use electron spin label. And we use the cross-relaxivity between the electron and the proton of water. And at the field that we are operating of 0.35 Tesla, this electron nuclear spin cross-relaxivity tend to be very sensitive to the right time scale, namely tens and hundreds of picosecond dynamics. And as I've mentioned to some of you already today when I met you, the spin label shouldn't be able to pick up anything authentic about the water unless water is forming is displaying network property. If water was individual, I have a spin label, then I have already modified the site. It shouldn't work. So when we started this study, it's not that I expected it to work, but it works. And the reason is that water has much more long-range properties than we imagined, okay? The second technique that we more recently got into is oxygen-17 NMR chemical shift. Turns out it's an extremely good experimental measure of electronic structure of oxygen-17 that is very sensitive to hydrogen bond lengths, as well as how many donor-acceptor pair it has. So it's an ideal technique to get at these structured waters. But I'll start with the dynamic measurement first, okay? So just maybe 1.5 slide on the technique of Overhauser DNP. For those, because there are many spectroscopists also in the room, as I said, it's about the dipolar interaction between the spin label, the electron on the spin label, and the proton of the water. At the end, the underlying property or the underlying physics is a dipolar correlation function that we cannot directly measure, can be computed. But this dipolar correlation function, its Fourier transform, is presented as the spectral density J of omega. And we can measure the amplitude of J of omega via T1 and NMR signal enhancement. Okay, so we are indirectly measuring properties of the correlation function. One more slide on that. And the only thing I want to emphasize is this is a cartoon version of the same thing I said, how dipolar correlation is measured. But the only reason I want to show you that is the following. So there's electron spin. I flip it, and then only water that moves at a certain speed at a certain distance will actually also flip, move away, and then live very long because T1 is very long. The main point is this. Even though I'm measuring bulk property, what I'm measuring in terms of enhancement has to come from direct contact, dipolar contact of the proton spin with the electron spin. So that's why we are measuring local property within less than a nanometer, okay? So I think I have to just go through this. Okay. And just to close the loop. For a long time, we didn't know oxygen 17 chemical shift could be such a powerful tool. We only started doing that about maybe a year and a half ago. But till then, what we did is we measured the dynamics and knew from our computational friends that, for example, diffusivity and p-tet, meaning tetrahedrality, correlate beautifully. So we now knew that water dynamics that we measure, which is equilibrium dynamics, correlate both with water structure, as measured by this tetrahedrality, as well as entropy properties. So we are at equilibrium, sensitive to thermodynamic properties of local water. Okay, so now back to this cartoon. I would like you to think about, if I have a surface of the type 1 and the type 2, how should the dynamics now look like, right? In the first case, the more hydrophobic sites I have of the sort 1, the slower the dynamics should be, right? Because I'm ordering the water more and more. In the type 2, the more of the hydrophobic surface I have of type 2, the faster the water dynamics will become, right? Because in the first case, I'm ordering water. The second case, I'm generating more disorder, right? So we were able to actually demonstrate that in one of my favorite studies that we did. And I think I'm just going to go all the way through the cartoon. And this is work we did many years ago with my colleague Susanna Scott when I was at UCSB. Their group generated silica that in one case was inorganic silica heat treated at different temperature to control the hydroxylation level. And in that case, we controlled hydrophobicity as measured by polarity measurement, but it's a macroscopic scale hydrophobicity, right? So the diffusion dynamic increases as the hydrophobicity increases. But in the other case, they prepared organosilica by adding more and more phenyl or biphenyl group. Those are nanometer scale hydrophobic sites. So we have more and more of this water ordering as reflected in the diffusivity measured by ODNP going the opposite way. But this was a demonstration that these sort of properties can be measured, but then we drew some hideous pictures like that. And of course, hopefully we'll get to the real structural property with other measurement. So now I have to go back to my tau monomer, okay? So what is the prediction then? If there is such a thing as water-mediated pinning, I should be able to see the hotspots on the tau monomer, and I should see it on a single spot. And that spot should also be the one first reacting, right? Let's see whether that's what we see. So what you're seeing here is experiment and theory. Experimentally, we indeed see in this peptide that's active and making a prion, that only the site around 300 in the middle is dramatically depressed in terms of diffusivity. And that's also the site that computationally around site 300 and 301, where my computational colleague from Professor Joan Shea and Scott Shell group and Dr. Saeed Najafi did the work, that both the tetrahedrality was the greatest and the de-wetting solvation-free energy was also the lowest, easiest to de-wet. I think this is shown here with a calculation called Indus calculation. They're pretty involved computational studies. So now the question is, with that, what should be the prediction? So if the site that harbors structured water is the pinning site, then we should see the monomer docking on the pinning site first. So let's think about whether we can prove that first. So here's a cartoon. So what we did is we took monomer, isotopically labeled, but we isotope labeled only the valine. It's only 20 amino acids, so we can purchase it. Okay, that's nice. So we isotope label the monomer, we do solution NMR, and we add our fibers C that act as prion, meaning they recruit my monomer. So what should I see if pinning is happening? then site 300 or 301 should disappear first, because as soon as my monomer become immobilized, solution in a mass spectroscopy will broaden the line because it needs tumbling. So we could see by the disappearance of the NMR signal whether we have successive pinning. And this is, of course, imperfect experiment because it's the initiation is done by mixing. Initiation is imperfect, but nonetheless, could we see that? So first you see all the assignments, and indeed, site 300 changes first, then it disappears. 301 becomes a lot smaller, and then 308 and 309, which is kind of peripheral, but still in the more hydrophobic region, and ends almost do not change until much later. Okay, so we see that the successive pinning is indeed working. So this was very exciting. So V300 initiate the pinning. Then my colleagues also did computation with this de-wetting free energy calculation. Just for your information, you cannot calculate or compute or even rationalize hydrophobicity by thinking about the amino acid makeup. Probably it's obvious to you, but I'll just restate in case it's not clear. Because hydrophobicity is determined by what water sees, it's not obvious that a valine is typically hydrophobic, but it's not obvious whether the site is hydrophobic, more hydrophobic, less hydrophobic by just looking at an amino acid makeup, because it's context dependent. So you see here a map, and indeed we see the red, which is then more tetrahedral as well as more hydrophobic, being focused around the site that we had experimentally seen. So, okay, I have time, right? Okay, so I have two things to do still. You probably noticed that I've convinced you probably that this is a real mechanism, but I have not really shown you the direct signature of the structured water. So that's one of the things I want to do. But what I want to briefly talk about is, okay, pinning works, let's say. How does it matter? Well, it matters because we think once we know that that's a key mechanism, we can control how peptides come together and where, especially if we know how to install a hotspot and move a hotspot. So that's now our next work. And I will tell you one of the discoveries we made, which is completely counterintuitive, that I will make a case why phosphorylation of all things might be one way how we can install a hotspot. Of course, it's not the only one. There are many, many other PTMs and mutations that can probably do that. But because it's so unusual and because it involves the thinking about water, let me talk about that just a little bit, okay? So we wrote one of the most unusual papers that we didn't intend to write, which is that phosphates assemble under all biological conditions. And the reason we discovered it is we wanted to make calcium phosphate cluster. That was the goal. And my students weren't able to add calcium for 1 year because the NMR signal of phosphate did something opposite to what it should do. It was broadening with increasing temperature instead of narrowing. Lines should narrow with more tumbling, not broaden. So it didn't make sense until we understood that they form assembly. When you increase the temperature, what happens is phosphate is hugely hydrated. And when you heat up or you add the certain Hofmeister ion or you add glycerol, you add PEG, there are many ways to do it. You can steal water from the phosphate group, okay? And then it basically promotes hydrophobic association. And we were able to measure that the phosphate is losing its water by measuring the hydrodynamic radius as a function of temperature. So the consequence of that was, once we understood that phosphates can cluster We then also tested, and indeed phosphoryl groups also can cluster. And furthermore, phosphoryl groups can form 1D wires, but not covalently bonded. Hydrogen bond mediated, water mediated hydrogen bonding, okay? So here I'm not showing you the water measurement, but what I'm showing you is that it was always a mystery to many of us. Why is phosphorylation associated with neurodegenerative tau proteins if they're negatively charged and therefore should somehow not be good for aggregation? But all pathological tau have phosphoryl groups in them. It's a hallmark, many, right? So it turns out phosphoryl groups love to line up because they form water-mediated wires. How do we know that? Number one, the stability shoots up. The quality of the fiber shoots up. They're very potent. And we can do something called multi-quantum NMR to show that we can induce up to 6 coherence excitation to show that phosphoryl groups are dramatically, regularly ordering. Okay, so we made, I think, tangible progress towards replicating atomically precise fibers. And the challenge that remains is that we can at will control assembly and the product that comes out of it. So there's a lot more work to do. So, yeah, I think I'll spend 5 more minutes, okay? So the most important part that I haven't talked about yet is I gave you a lot of circumstantial evidence because that's how we got to this point. But I haven't shown you the direct spectroscopy signature of the 2 types of water. So one way to illustrate that is to go to actually dimethyl sulfoxide, okay? And the reason is, if I take dimethyl sulfoxide that you know is very miscible with water, and you would think, what else can we learn about dimethyl sulfoxide? Well, it's a very nice model system to think about because the sulfoxide group strongly hydrogen bonds water. And the 2-methyl group obviously cannot directly hydrogen bond water. So what is water supposed to do? Why is it so soluble? Well, it turns out by experiment of terahertz and our own measurement as well as computation that water forms this structured, what my colleague Martina Havenit calls tetrahedral wrap water around the methyl group and directly bond water on the sulfoxide group. So these 2 types of water or 2 structures of water is very nicely represented in the dimethyl sulfoxide, and you can imagine what it means. But another point I want to make is that is seen with any solute. The only solute that may be an exception, I don't know, is sugar, because sugar is weird, okay? But other than sugar, there is no solute that can fit into the water structure perfectly. If it cannot fit perfectly, there will be hydrophobic hydration, because at some point, water will run out of angle to do something with the solute, so it has to wrap around. So indeed, even glycerol that you think of as very hydrophilic still is hydrophobic, depending on the conformer and location. So it also has 2 types of water or more, and so does any solute, okay? So, well, the p-TET and the dynamics correlate beautifully. As you see, those are experimental measure on the y-axis and computational measure on the x-axis. And then we did a shocking experiment. After that, I told my students, I almost apologized. I said, I'm sorry to be asking you this, but could you please take DMSO water and glycerol water, just sprinkle some oxygen-17 isotope labeled water in them, make the whole concentration series and measure, please, the 1D NMR spectrum. Why did I apologize for it? Because it's such an obvious experiment that I thought somebody must have done it. And if it would have worked, it must be stupid. It cannot be, right? And so what was I looking for? I was looking for, do I see a change in shift? And we couldn't find anything in the literature. And so what do you see? As a function of concentration, my shift bends. What does it mean? It's relative to bulk. It goes in one direction and the other direction, okay? There are 2 types of water. They are, when you run out of water, eventually, which water should run out first, okay? You first will lose your wrap water, and eventually you will lose your bound water, right? So when we saw this band, it was shocking. And then I told my students, cannot be that nobody measured this. How can this even be? So it turns out there is a paper, but they concluded that oxygen 17 with is very sensitive to viscosity. That's a well-known idea. But they measured up to 40% and concluded the shift isn't changing. Well, up to 40%, it's hard to see. If you go all the way, it bends over, okay? And one of the main reasons why I think those experiments have not been done much is because the assumption always was, well, water is not that different and it exchanges fast. What can somebody possibly see? But obviously the chemical shift is a very good measure, okay? So now, almost done. I'm sorry, I didn't mean to go too much over, but so we then thought, okay, we don't know quite how to do this oxygen 17 measurement site specifically. We are working on it. It's not so easy. But then we said, okay, let's measure the whole ensemble. Why not? We take a relatively high concentration of the monomer and the fibril, and could we see 3 populations? Because there should be bulk, and there should be wrap, and there should be bound. So we see this weird feature. It looks like it could be. The technicality of it is a little bit tricky, so it takes a little bit of time to explain, but you could see how this could be 3 population. And we see that on a protein that I think I won't talk about, ASLOF2 as well. But the most important part, and the reason I'm showing you that is because this measurement we couldn't do on tau, so I'm showing it on another protein. We see the same feature on many proteins. So when you take the left, middle, and right features and filter, the T1 relaxation time are different, very distinct for all 3. So I'm making a case that we have distinct population, even though the spectroscopic resolution isn't great, okay? So I'm coming to an end. So I think, of course, I think I just have to skip the story. You have to ask me later, what do I mean by hydraulic pump? That's more of a later conversation, but I'm certainly happy to entertain you. Okay, this has to just go. But I did want to show one more thing. So I hope I have given you an idea that the development of experimental tools were really key to showing, with a lot of patience, this idea about the active role of water. But I also want to talk about, briefly, one slide, being at UW, I have to talk about Gerald Pollack when I talk about water. I have to, right? So you know that he has written this 4 Says a Water book. And the issue is, or not the issue, but the thing is, I know the water community quite well. And I used to go to the Gordon Conference of Water every year. Then I go still regularly. But they are completely non-overlapping with this other community, but that's a pretty big community. So whether there is something to it or not, or something in between, there is almost no overlap between spectroscopists and physical chemists, and then the biomedical engineers and so on that talk about this long-range ordering of water. I think there is something to it, but there is almost no spectroscopy done, so hard to understand what it actually means. But the reason I am showing this is that I think we should, and perhaps are ready to venture into having the tools ready to look at this long-range ordering, because some of the claims are quite wild in the sense of long-range ordering and even different kinds of structures. I'm just going to say what the claim is, okay? So I've shown you tetrahedral water and icosahedral water. That already took us a decade to prove. Now we are talking about charged hexagonal water. I actually asked Ken Dill, who is the author of StatMech book, right? And I said, do you think that's crazy? And he says, oh, no, we see that all the time. I said, oh, really? So I don't know the truth because we haven't measured it, but I think it's worth venturing into these questions too. So I took more time than I meant to, but I hope you got something out of this talk. And I will acknowledge several of my collaborators. Tau, actually, Professor Ken Kosick got into this tau business. Initially, it was meant to be a model, and then they got really serious about it. many people, especially Scott Shelses and Scott, for measurement of water and my students, and thank you for your attention.
Thank you very much. Wonderful. Questions? Thank you, Songi, for a wonderful talk. Any questions? William.
Mm-hmm. I wanted to escape the presentation, but I don't know how to do it. So I'm just going to flip through. Hopefully it will come there fast. Sorry. Okay. Dear, come on. Where is it? Yeah. How do I escape this? I don't know. Okay. Can I escape it?
You should be able to see all the
Yeah, screen.
by title. See if you can see
By title. Oh, I see. So maybe at least I can go way earlier and then from there flip. Okay, that should be, I'm pretty close to it now. Okay, I'm there. All right. Mm-hmm. So this is now dangerous territory. As you probably know and Stefan knows, DIA is the most difficult to apply to a disordered ensemble because you have short to long distances. There is so much uncertainty in the interpretation of P of R. What I can say is the analysis, we have a few options. The decay curve doesn't have enough shape and feature to probably answer that question. So there's one way we could do it is we could convince ourselves with more experiment that there are 3 populations and do basically a principal component decomposition and then talk about population. But the problem is we are getting P of R of something that has so many oscillation overlapping. At the end, it looks like kind of a crooked exponential decay, okay? And we are doing a ill-defined transformation to get a P of R. So that's a difficult question to answer. But what I can say is because we do this experiment many times and in triplicates, that the trend is real and that we have 3 populations-ish.
Testing that these then undergo a lateral association as well.
So first, what I'm saying is in polyphosphate, my understanding, and please correct me if I'm wrong, there are stronger association than just loosely water mediated. Am I right about that? I think in polyphosphate, they are both covalently bonded polyphosphate, but also probably hydrogen bonded, but they are much stronger associations. So the clustering that I was mentioning in the first part was very reversible, completely reversible cluster formation, which still has a significant water cloud around it. In the case of the phosphoryl group, what I'm saying is they align in high order, but the spacing is about 4.8 angstrom, and that needs to be at least probably 1 to 2 water layer in between the phosphate. So I don't know what I'm answering your question exactly.
This happens in cells. You discovered apparently monomeric section, but polymers along the length, polyphosphate polymers. This test appears to be related to a mechanism for sort of go out and harpoon, pull back pieces of DNA to the.
It's extremely interesting. I cannot exactly answer that question. I know and noticed that bone formation, DNA, all sorts of processes rely on assembly of phosphate of some sort. I think the polyphosphate formation is stronger and more covalent interaction. In this case, what I'm proposing, because I know from the distance, it's not a polyphosphate distance, a 4.8 is still a large distance, which still it is sandwiched between water. I am suggesting, though, that in the phosphorylated peptide, that the phosphoryl group alignment comes first. I am indeed saying that. And that organizes peptide. And then once it comes closer, there are other forces that induces shape, folding and a beta sheet formation. Yeah, phosphate is really interesting. Uh-huh.
The first step in that polarization is backwardization, and then studied by
I would love to hear about that. Yeah. Okay. Yeah. Well, what I can say is that we looked at phosphate assembly at pH 2 to 10. And even though the extent to which they form association differs, all of them do that. So H2 PO4 minus, all the way to PO4 2 minus, they all do that because of this extensive hydration shell. And I think we don't know enough about it, but there must be something special about these phosphate and phosphoryl groups because they're so abundant in biology whenever it needs to make extended structures.
Any other questions? Munira.
Mm-hmm. Glycerol and DMSO, yeah. Mm-hmm. 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 Yeah. So we did the oxygen 17 experiment first on DMSO and glycerol because there we could do rigorous analysis for computation too. But we did that on all sorts of proteins. So in tau and the ASLOF2 that I didn't show, we see these three populations. And the part that I could have shown but I didn't is so this protein that I talked about, how it's light activated. And so it basically, like a water gun, squeezes water, retracts, squeezes water, retracts. How do I know? We took the oxygen signature, turned on the light, off, on, off. And we can see that signature, blink, come back. We repeated it 10 times. So I can say that these signatures are showing up in protein, even though we measure the bulk property. Now, we would like to measure that size specifically, but that's quite a bit more tricky. So, interconversion between which one? So I think we need for that simpler system. Because even in DMSO, I can tell you that the electronic structure calculation that we are doing, there are many contributions. We can add it up and we can recreate the experiment only because the local contributions are so local that they're additive. So we are lucky. But still, we are talking about a vector that points in all directions and we have to add it up and we are lucky that it's here. But I think we need simpler system for that. And probably, I think Anne suggested that. So this sort of gas phase spectroscopy is needed to have phosphate with, or any simpler system where we can actually see what happens if I hydrate phosphate with 1 water, 2 water, or 2 phosphate with 3 water, 4 water. I don't think we can do that with that. We are, in NMR, we are still talking about 10 to the 16 molecules. That's a lot of molecules. Yeah.
So if there are no further questions, let's thank Songi Han again for a wonderful presentation.
Thank you. Thank you.
And there's a reception after this that is happening in CHP 102, if I'm correct. All right, see you there. Thank you.